Hi, my name is Blessing Edje, and I work with the Water Quality Division of DC Department of Energy and Environment. My presentation today is on stream assessment, benthic macro-invertebrate collection, and identification. This presentation will focus on introduction of benthic macro-invertebrate, how samples are collected, how we conduct stream assessment, how samples collected are processed, how we identify the benthic macro-invertebrate, and preparation of slides. What is benthic macro-invertebrate? Benthic macro-invertebrates, they are organisms that lack backbone, and they can be seen with the naked eyes. However, there are some features of these organisms that cannot be seen with the naked eyes. To view and identify such features, we use the stereo microscope. The word benthic means bottom. Hence, from the name, these organisms live on the bottom of streams, ponds, rivers, lakes, etc. These organisms are very important in the food web in aquatic system, as they serve as both prey and predator in the food web. The basic principle behind the study of macroinvertebrates lies in the fact that some macroinvertebrates are sensitive to pollution than others. For example, the larvae of mayflies, stoneflies, or even caddis flies can only survive in clean water. Consequently, it is presumed that the presence of these organisms in a water body is an indication of good water quality. However, it is not certain that the absence of these organisms are indication of poor water quality due to other factors like low dissolved oxygen, which results from excess nutrients, water flow, salinity, pH, temperature, habitat loss, and many more factors that can hinder the survival of the organism in a water body. We collect our benthic macroinvertebrate sample in the spring season, usually from March to May. On getting to the stream, 75-meter stream range is measured, beginning with the 0-meter mark at the downstream end of the stream and ending with the 75-meter mark at the upstream end. We are required to mark 4 locations: 0 meters, 25 meter, 50 meter, and 75 meter within the 75 meter with a spray paint or flags. Our samples are collected randomly from 20 points within the 75-meter range using a 500 micro-merge D-frame net. Environmental parameters such as pH, conductivity, temperature, and dissolved oxygen are measured using wire-size sun instrument at the stream. Samples collected are strained using a 500 micro-merge strainer to remove any excess debris and kept in a glass jar containing 95% ethanol to prevent the samples from deteriorating. 2 waterproof labels are filled out, like in the picture here. 1 label placed inside the jar and the other taped on the outside of the jar to ensure the label is properly secured. The benthic macroinvertebrate matrices are used to determine stream health based on an adaptation of the 2007 Maryland Biological Stream Survey. Stream assessment. Under stream assessment, we have the habitat assessment guide sheet that help us to do the stream assessment whenever we go out for a sample collection. We have different parameters, and also we have rating numbers that we use to rate the stream. For example, we have the optimum, which has numbers 16 to 20. We have suboptimal, number 11 to 15. We have marginal, 6 to 10, and poor rating is 0 to 5. The parameters we look at are the in-stream habitat, and we look at the second parameter is the epifaunal substrate, what kind of substrate that is available for the organism. Is the hard substrate, is the substrate suitable for the organism? We identify that and we rate it based on the numbers that are on this guide sheet. The third parameter is the velocity depth diversity. For the velocity depth by diversity, we rate it based on a variety of velocity depth regions present at the site or at the stream. For example, do we see slow to shallow? Do we see slow to deep, fast to shallow, and fast to deep? So whatever we see there, based on what we have on this sheet, we rate it and we give it a number. The fourth parameter is the pool glide eddy quality. We rate this based on the variety and spatial complexity of both slow or still water habitat within the sample segment. And the fifth, we have the refill run quality. We rate this based on the depth, complexity, and functional importance of the refills. How many refills do we have there? What are the qualities? And we'll give that, assign a number using this sheet. Number 6 is the embeddedness. Embeddedness, we use percentage. We don't use the numbers, but we just use percentage to rate the embeddedness. What is the composition of the material of the stream, bottom of the stream? Do we have fine particles? Do we have gravels? Do we have cobbles, boulders in the stream? So we evaluate that, the percentage of what we have, and we write that down. Number 7 is the shading. Is the stream covered fully with trees, or do we have like the stream fully exposed to sunlight? So we also calculate that percentage of shading that we have on the stream, and we write it down. The last one here is the trash rating. So we look at the amount of trash present within the stream, and we evaluate and rate it based on the number present here on the sheet. Here we see this slide contain the sheets we use to record what we see during our assessments. We have the sites, the dates, include the dates that we went there for assessment and the time of assessment. I have 3 points here that I want us to quickly look at. We have the first one here is the sampleability. We have the benthos, habitats, and water quality under sampleability. What does simply require us to do? We check the box if these three are done. If we carry out this 3 operation, we check the box. For the benthos, it means you actually collected samples from the site. For habitats, means you did habitat assessment. You assess the stream and you check the box. For water quality, you took the temperature, the pH, and the different parameters of the water quality. You check the box here. But sometimes we are not able to go into the stream, so you only do the habitat assessment and the water quality. And some factors that can hinder us from not being able to go into the stream are maybe the depth, the turbidity of the water. So that can prevent us from going into the water, or maybe the water has a high flow. That can also prevent us from going into the water. So we only do the habitat and water quality. But on this sheet, we'll make a note stating the reasons why we are not able to get our samples. Next here is the road covered. So for the road covered, we look at the roads that are present close to the stream. We take measurement of the road close to the stream. And if there's any coverage on the stream, we take the width and also the length of the coverage. I'm going to show you guys what that is, a picture of a covert in a stream in upcoming slides. Other parts I will look at is a benthic habitat sampled. What kind, the 20 points, like I mentioned earlier, that we are going to pick to get our samples from. What kind of habitats are they? Are they riffles? Are there riffles? Are there root words? Are there leaf packs, macrophytes, or undercut banks? So whatever points we pick to take our samples from, we're going to put it down on this sheet of paper. So here are pictures of the root coverts, the riffle, and the root word. So here is a picture. So this middle here is what the covert is. This is the covert, and we'll take the length of the covert and also the width of the covert. We'll put it down on that on the previous sheet that we're seeing. So riffle. Riffle is a portion of a stream with the fastest flowing water. So we look at how many points here do we take our samples from. Sometimes in a stream, you only see riffles. Sometimes you don't see, you see less riffles. Sometimes you see root words, you see leaf packs. You see a variety of these habitats, but sometimes you don't. So whatever you see, it just all depends on the stream you're getting your sample from. And here's a picture of the root words, the roots of trees by the stream of the bank. The root must be submerged, must be in the stream before you classify it as a root word. Sometimes there are some roots that are close to the stream, but they are not in the stream. It has to be inside the stream for you to be able to classify that as a root word. Here we have the pictures of the woody debris, the leaf pack, and macrophytes and undercut banks. A woody debris includes any trees, logs, branches, roots, words in the stream. So like this picture here, we see the tree, fallen trees. Here we'll consider that as woody debris. So take note of that and put that down on our sheet. It is, yes, the leaf pack. So we also take samples from the leaf pack. We just rinse the leaf while we take the samples. So we, under the sheet we saw earlier, we marked that down that we took samples from leaf pack. And here is a macrophyte, picture of the macrophyte. And we have some floating plants, and we have some emergent plants. We look at what we have in our streams, and we see if we want to take our samples, if that is where we want to take our samples from. And here we have the undercut bank. Undercut bank occurs as a result of soil erosion. We also take notes of that and put it down on our paper. This is the second set of sheets that we use for our stream assessment. And we already talked about the trash rating. We have the riparian vegetation, the buffer breaks type that are present on our streams, the land use types that are present, are there any channelization? And this is the place, the portion where we actually write down the water quality that we do on the stream. Using our wide size sum, we take our water quality measurement like the temperature, the dissolved oxygen, the pH, conductivity, as you see on this sheet. So for riparian vegetation, I have some pictures here that will just guide us when we go out to look at the forest, what kind of trees are made of, what kind of trees are in the forest. Do we have the deciduous or coniferous trees? Do we have grass? Do we have lawns? Whatever we have close to the stream, we observe and also put that down on our paper. We also take note of the land use types. What kind of land use types do we have close to our streams? Do we see old field? Do we see wetlands? Do we see surface mine? Do we see a lot of residential areas in the close to the stream? Do we see commercial or industries? Do we see croplands? golf course. So here I have just 3 pictures, and the first picture here, A, is a picture of a wetland. The B here is a picture of the orchard vineyard, and the C is a picture of what a pasture look like. Buffer breaks. There are different buffer breaks that we are interested in when we go out on a stream survey. The storm drain, we have the tide drain. Is there gully around there? And are there raw sewage? In picture A, we see the raw sewage deposition in our streams. That is, we look out for that and put that on our paper. Channelization. Adhere channelization is our stream channelized. We look if there's any kind of channelization on the stream. Like the picture here, we have different type of channelization, but I have 3 pictures here for us to know what it looks like. Channelization is just the process of redirecting the natural stream in an artificial modified manner. So we look at for the if there any type of channelization present in our stream, we'll take note of that. So this brings us to our sample processing. Before we sort our samples at the laboratory, what we do, we rinse the samples with distilled water, and we place the samples in a grid tray for sorting. We sort out 100 individuals, and we identify them. And when necessary, we place the organism, we clear the organism using potassium hydroxide, and we slide mount it and observe it under the microscope using a stereo microscope to identify the organism. Before we go into the identification of Benthic macro-invertebrate, it is important to know that they are classified into 3 main phyla groups. We have the arthropoda, we have the annelida, and the mollusca. However, there are other phyla groups which include the bryozoa, the cnidaria, nematode, and so on. But for the purpose of this presentation, we're going to focus on the 3 main phyla groups. The first phyla group we're going to look at is the phylum Arthropoda, and the subphyla is Arthropoda. This group of Benthic macro-invertebrates insects are the largest of this group, and they consist of winged and wingless insects. Some examples of this of the insect in this group are ants, flies, cockroaches, butterflies, etc. One general features of this organism is the presence of 11 abdomen segments. In previous samples collected from DC water bodies in 2009, a total of 92,000 of macro-invertebrates were identified in 17 samples collected, as reported by Ritrom Associates Incorporated in their October 2010 report. According to the report, the most common Benthic macro-invertebrate identified in the system are the non-biting midges family in the phylum group Arthropoda and subphylum Octocladine. So we'll be looking at some examples of this group, this Arthropoda group. The first example here is the Chironomus. This Chironomus group, I have some arrows here to show us some features that we should look at for while we're identifying this organism. The first is the head part. The head part is sclerotized and is in form of a capsule. And also we see the mentum. The mentum here is broad, and it has multi-toothed combed. It lacks true legs, and mentum is present. The second example here of the phylum Arthropoda is the Tinamania. And here we have a general features. General feature here is that the antenna blades are sclerotized, and the body center is scattered and is long. Mandible has 3 inner teeth and 1 outer tooth. The third example is the damsel fly. Here we see that the damsel fly here has a very conspicuous eyes, and the wings are brown in color. The fourth example of this group is the mayfly, and we see the parentheses mark in the middle tagger of the abdomen. And also the tail end, we have the dark band at the beginning of the tail, in the middle, and at the end of the tail. The second phylum is the phylum Annelida. The phylum Annelida annelids are known as segmented or ringed worms. They are mostly aquatic, marine or freshwater, and some of them are terrestrial. We have some subphylum of Annelida. We have the polychaetes, oligochaetes. Polychaetes here denotes many bristles, and they are mainly marine or freshwater, while oligochaetes denote few bristles. Example is earthworm, and they are mostly terrestrial. One example of the polychaete worm is the Manayonkel speciosa. And this, the general identification hint of this organism is the head part of this organism. It has a branchial crown with a collar, and eye is present. Their body is made up of sandy membrane, and the body segment lack chitin. The first segment part lacks chitin or bristle. Here I have 3 examples of oligochaetes, the subphylum oligochaetes. And the first one here, we see that they have the paired bristle, and the second one also has bristle all over the body, and the third one lack bristle in the body. So these are some features we can use to identify or differentiate between some of these organisms while we look at them, or while we try to sort and identify them in the lab. The last phylum group is the mollusca. The mollusca, they include snail, clams, scallops, octopus, oyster, squid, and so on. And one general features of this phylum mollusca is that they are covered, their bodies are covered with hard exoskeleton-like shell. Example of this group, phylum mollusca, is the Scylla acute. And one main features for this organism is that the body is covered with brown colored, thick, strong shell that has elongated teeth. And also it has an opening that looks like a shape of an ear. This brings us to the last part of this presentation, which is the preparation of slide or preparation of wet mount. First step to prepare a wet mount is to get a clean slide like we have here. We had a drop of glycerol or distilled water, and using a tweezer, get your organism and put in the fluid. Get a clean slide cover like we have here in this picture. You cover your organism, wipe the side of the slide that has any excess water or any excess glycerol. Then you place your slide under the microscope and view your organism. Yeah, I have 3 questions for you guys to think about. And why, after watching the video, you think about these questions and get answers to these questions. First question here is, what is the basic principle behind the study of benthic macroinvertebrates? Second one is, what are the importance of benthic macroinvertebrates to stream health, water quality? And the last question is, what are the uses of stream habitats assessment? All of these I've mentioned in this video, so you take your time and write some answers, give some answers to this question. I have some link here for you to, if you want to check for more hints or more pictures of benthic macroinvertebrate, you can use this link to check for some of them and get familiar with them. Here are the references that I used in preparing the slides. This brings us to the end of the presentation. Thank you. I hope you enjoyed the presentation